Hello and good morning. Welcome to the latest installment of the Climate Justice and Resilience lecture series here at Evergreen. I'm Rachel Hastings. I'm a member of the faculty at Evergreen, and I'm excited to have you here. I just want to let you know that questions are welcome, and there's a Q&A feature that you should be able to find at the bottom of your screen where you can type in your questions, and those will be answered by our speaker at the end, probably at the end, although possibly as we go along. So to introduce the speaker today, we have Lalita Calabria, who's a member of the faculty at Evergreen.
Thanks, Rachel. Welcome, everyone. It's my pleasure today to introduce Dr. Rob Smith, who will be speaking with us today on climate change modeling with lichen communities. Rob Smith is a forest ecologist and ORISE fellow with the U.S. Forest Service's Air Resource Management Program in Washington, D.C. I've had the pleasure of working with Rob on a couple of projects related to bryophyte and lichen ecology, and it's great to have him here today. Let's see, what else do I want to let you know about Rob's background? He aims to guide forest management by forecasting vegetation responses to pressing global changes, especially where atmospheric stressors and disturbances intersect. Rob's work lies at the broad intersection of forest ecology, biogeography, and high-dimensional statistics. He contributes to the National Atmospheric Deposition Program, an interagency cooperative which tracks atmospheric conditions and their environmental effects. He was a researcher previously at the USFS Forest Inventory and Analysis Program in the Pacific Northwest, and he's taught botany courses at Oregon State University after earning his PhD there with a focus on detection and prediction of climate change signals using lichens as bioindicators. Welcome, Dr. Smith. I'm looking forward to hearing your lecture, so let's get started.
Thank you very much for that kind introduction. And thank you also for the invitation to be here with you today. It's an honor. So I will share my screen here if I can certainly do so. So hopefully you have a full screen there. So what I hope to share will be talking about modeling lichen communities to detect global change signal. So the modeling is sort of the tool, but the ends to the mean of detecting climate change and other kinds of global change signals in vegetation. And so my overarching question, the motivation for that is asking, what is the fate of future forests? What will future forests look like in terms of their vegetation when they are subjected to changing regimes of climate change, of air quality and air pollution, of disturbances that are departing from historical ranges of variability? So the question there is, how will vegetation respond to that? And that's a question that's important for not just determining things for the vegetation's sake, but also for its ability to provide things to us human communities and to other kinds of forest life. So altogether, that means that we need to have the capacity to look forward, to anticipate and forecast into novel climate spaces that this vegetation has perhaps not seen in thousands of years, if it's seen this kind of change at all. So looking for that forecasting capacity, that's what motivates me. So I focus on epiphytic lichens, and I would like to make a claim, maybe it's a bold claim, that they are the world's best climate sensors. And I say that because they are distributed practically everywhere. They are free. They don't cost anything. You don't have to put a battery in them to run them, if we can detect the signal that they're trying to tell us. They are climate sensitive, and that's something I hope to demonstrate today. You can transplant them, you can manipulate them and experiment with them, all in a tidy little package. So the fact that they're distributed fairly ubiquitously across forest vegetation makes them useful to us. I should also stop and mention, when I'm talking about an epiphyte, I mean an organism that grows upon another plant. So epiphytic lichens grow harmlessly upon the bark and outer surfaces of trees and shrubs. So there's also the intriguing possibility that changes in lichen communities might precede changes in other kinds of forest vegetation and forest processes. So they have long been used as indicators of air quality. They're known as the canary in the coal mine for air pollution and air quality. And I'd like to see if we can use them in the same context to be bioindicators of climate change. So when I say lichen, I am talking about a photosynthetic partnership. So a lichen is a fungal partner, a fungus that makes up the bulk of the body of the lichen, and also which determines the name. It's the species identity is the fungal identity. And the second partner, or sometimes even a third partner, is a photosynthesizing partner, typically a green algal component, an algae that's just making a thin layer, and if you were to take a cross section, that actually does the active work of capturing the sun's energy and taking up carbon dioxide to convert into growth, to convert into the body. So lichens are a photosynthetic partnership. We treat them more or less as we would treat a plant. They have a plant-like growth form, but a lot of unique capacities that make them quite different than plants. For example, they have an atmospheric like lifestyle. They live essentially exposed to the atmosphere, growing on the outsides of other plants without protective bark or cuticle or outer structures that would buffer them from atmospheric variation. They don't have roots or anything that would allow them to tap into soil moisture or soil nutrients. They get 100% of their nutrients from the air, from atmospheric deposition, and from moisture that's in the air. So because they're decoupled from terrestrial processes, they're effectively atmospheric, and they should respond closely to atmospheric variation. So overall, I mentioned earlier, my goal is to detect and also predict with a forecasting ability global change signals in lichens across multiple scales. And what I mean by that is from the individual lichen all the way up to collections of multiple species that we call a community. And more than that, looking across landscapes. So what I will hopefully share with you briefly today are 4 different stories at different levels of organization or different scales, from the individual to the community, combining both experiments where we manipulate climate conditions to be able to attribute changes to climate, to forecasting kinds of inference, and then actually looking at observed changes that happen retrospectively. What has already happened in terms of changing communities? That's what I hope to talk about today. So the first issue, how will individual physiology or the performance of individual lichens change under simulated future climates that we expect? So I'm part of a larger experiment in northern Minnesota called the Spruce Experiment. This is a whole ecosystem warming experiment where these open-top enclosures are totally encapsulating the tree canopy in a black spruce bog. And so the idea here is that by adding combinations of CO2 to emulate future CO2 conditions in the atmosphere, and adding warming both above ground and from below the peat in this peat bog, that we are simulating a range of potential future climates. The chambers are controlled to basically to every second to maintain a certain differential temperature relative to the ambient temperature. So if it's -10 degrees in the winter, then the highest treatment is 9 degrees Celsius, so 9 degrees above that. And even if it's in the middle of summer, say it's 90 degrees Fahrenheit, well, it will be quite a bit warmer than that in those chambers. And I have been in these chambers in August, and believe me, they are quite warm. So what we have done is used lichen transplants that we've installed in the chambers at the different levels of warming, ranging from 0 to 9 degrees Celsius above ambient. And then we take the transplants and look at their growth and their physiological responses. So here's my colleague, Elisa DeMaglio. She's installing the transplants, one of which is in detail in the upper right. I think of the transplants as a little lichen earring. It's basically dangling on the branch that you can safely take on and off the branch every year to make your measurements and then replace it for long-term repeated sampling. So we have tracked these individuals every year since 2013 when they were first installed. And then 2015 is when the warming treatments were initiated. So we have 2 years of baseline measurements. What we find is that just the first year of warming that was applied, that is from 2015 to August of 2016, we see a rapid decline in growth. So I'm showing you here on the vertical axis, mean annual biomass growth, that's expressed as a percentage across an increasing warming gradient. So where ambient temperatures are down here around 0, individuals tended to have growth that was on average about 15 or 20% growth annually. But by the time you get up here to 9 or a little bit higher degrees Celsius above those ambient temperatures, we see almost a complete reversal of about 20% loss of the mass of this lichen. So the lichens at the highest warming treatments are essentially wasting away. And the idea is if we can take physiological measurements that help us attribute that to carbon balance, so carbon uptake, that will help us make that attribution. So with colleagues at the University of Minnesota, we are taking physiological measurements using chlorophyll fluorescence. This basically means you're taking an image of the entire lichen to look at the way that chlorophyll is active or is not. So we take measures like FVFM. This is just a measure of photosynthetic efficiency, how well photosynthesis proceeds. And since we're taking an image of each individual lichen, we can also get an estimate of the photosynthetic area of that individual that is actually doing the job of photosynthesis at the same time that we're measuring gas exchange of carbon. So we have multiple independent measures of photosynthesis that we're looking at. And these measurements were taken 4 and 5 years after the treatment, so in 2019 and 2020. What we find from these physiological measurements is that the warming treatments lead to something that we could call carbon starvation. So for both of these images, if you have points above the 0 line, you have a net positive carbon uptake. And below the line, carbon is lost from individuals. So with dark photosynthesis, we're essentially looking at respiration, so losses of carbon, which is normal even for the ambient individuals, ambient treatments, as well as for the most warm treatments and everything in between. So respiration is a sign of growth, but it can also be a sign of decomposition. So it's present throughout the spectrum. But the important part is to look at the balance during the light reactions of photosynthesis. So during active periods during the daytime, where the ambient individuals can make up for these respiratory losses by grabbing carbon, by performing active photosynthesis, but not so when you get toward the warmer end of the spectrum. So carbon starvation, these warmer individuals cannot make up, they are in deficit, they can't make up that carbon balance. So this pattern is attributable to decline of the photosynthetic partner, the photobiont. What I'm showing you here is the measure of photosynthetic efficiency called FVFM that you can see declines precipitously above about 4 or 5 degrees Celsius above ambient. Likewise, the active area of that individual declines pretty quickly beyond that point. So this tells us that the photobiont partner that's involved is struggling to do its job. It is at least as climatically sensitive as the fungal partner that houses it. Another issue with the photobionts, if we sequence their DNA to attribute them to different genotypes, what we see across the warming gradient, again here from left to right, increasing warming, the proportion of each of these green algal genotypes that's represented changes from one dominant genotype in the ambient and control treatments to a gradual loss of that genotype and gain of alternative ones. So there's this idea out there in the lichen world that photobiont switching, acquiring different genotypes of photobionts, if those novel genotypes are climatically adapted, that the overall lichen organism can also adapt and occupy novel habitats. So if this changeover were successful, and if photosynthesis and growth could occur, that might be a means by which lichens could persist in increasingly harsh environments. But for now, this is to be demonstrated a little bit more. So I'll pause for a minute. What I just mentioned there is individual performance, but we'd like now to scale that individual performance to anticipate how collections of multiple species in communities might shift or change. So how might the community composition change, and what is its vulnerability to change? So we're very fortunate in the States that we have systematic nationwide surveys of lichen communities as part of the Forest Service's FIA program, Forest Inventory and Analysis. So there's upwards of 9,000 or 10,000 surveys nationwide that have all been measured using the identical measurement protocol. They're all measured in exactly the same way. In a way, for each of these sites shown in little black dots here, you get a list of the complete species that occupy that site within measurement quality, within measurement tolerances. So there's a 2-hour cap, a 2-hour time limit to the survey, but you're going to gain upwards of 90% of the species that would occur there. I should note, you'll see clusters of plots in a few areas, specifically the Northwest, that I'll talk about momentarily, that have been intensified measurements as part of the Air Resource Management Program that I currently work for. So we can use these surveys that cover a broad range of climates, a broad range of air pollution conditions, to tell us something about where the species occur with respect to those environmental drivers. So for that, that's the approach that we use to quantify something called vulnerability. So vulnerability says how susceptible is a community to changing, to gaining or losing species over time. So we use the FIA data I just showed you, and for each single species, we can get a map of its distribution across the United States. And if we know for that species where it occurs geographically, we can map that distribution to its environmental distribution in the center here. So this is just a frequency of all these populations with respect to environmental drivers. Say this was a thermal gradient or a climatic moisture gradient or an air pollution gradient. Those populations that occur at or above the 95th percentile, we simply call them vulnerable populations that we can then map back out into geographic space. So for each single species, we know where its vulnerable populations are. But again, we want a community measure that aggregates across species. And that's what, if we take something that's just as simple as the percentage of species that occur at that site that are vulnerable, we get an index of thermal vulnerability that I've mapped here for those lichen sites in the Western US, where cooler colors are relatively less vulnerable. So in other words, you wouldn't worry about much change happening in those purple sites if climate change were to proceed. Whereas the hotter colors here indicate relatively higher thermal vulnerability. So for example, in the Central Valley of California, Southern Oregon, around the margins of the Columbia River Basin, the interface here between the Sonoran Desert and the Columbia Plateau. Those are places where we anticipate community changes to occur first under warming atmospheres. So this thermal vulnerability index is useful. This is one snapshot in time. What happens if we look at vulnerability to community shifts under future scenarios? And maybe it's not too surprising just based on the definition of vulnerability that I've told you, but levels of increasing warming under future scenarios will drastically increase vulnerability. So for warming scenarios on the horizontal axis increasing here, the percentage of sites in the US that we've sampled for lichen communities that we deem very vulnerable, I think here we used a value of 75% as the cutoff. We find that at the highest levels of potential warming, that basically 1 out of every 3 lichen communities in this data set is considered very vulnerable, where 3 out of 4 species that occur at that site are susceptible to change. So that's an interesting observation. Another way that we can slice and dice this from the geographic pattern is where, with respect to climate, is the highest vulnerability occurring. It's occurring at the hottest and driest extremes at the southern latitudes. So the lower latitude, the more southerly sites. Again, you can see that on the map. And also the lower elevations, so the valleys essentially, where the montane sites are not necessarily constrained in the same way by warming in this case. So that may be a little bit counterintuitive because a lot of people are expecting the cold tolerant species, the mountain species, to change first. But what we're finding here is that the places that are occupied by species that we assume tolerate hotter or drier conditions are the ones that may actually be changing most rapidly. We can also do the same vulnerability exercise for multiple stressors, not just our climate vulnerability that I've shown on the left, but also for air pollution vulnerability, for example, nitrogen deposition, which is at high levels is considered a pollutant. So reactive nitrogen has, as I mentioned earlier, has had a long association with lichen communities. And here we're looking at the intersection of air quality with climate. So this can be important for looking at places where multiple stressors intersect. For example, looking at the Central Valley of California, glowing in both of the maps here, telling us that there's high vulnerability to multiple stressors. And another way to think of that is there's multiple different avenues by which communities could change, given multiple kinds of global change drivers. Again, a brief pause. So what I've just shown you is forecasted or potential community changes. Now we can ask the question about what changes have we actually seen occur in the region of the Pacific Northwest over a period of 2 decades. Can we detect a climate change signal from the vegetation changes? Again, we're fortunate in that we have a long-term monitoring network of permanent plots in this region established by the Air Resource Management Program of the Forest Service. So for these plots, they are arrayed on a fairly regular grid, again, with some intensification, especially in the Columbia River Gorge. So I'm showing you plot locations, and the gray map in the background here are the counties of Oregon and of Western Oregon and Southern Washington. This white line here is the Columbia River bisecting them. So the points here are colored by the time that has elapsed between repeat visits. For each of these sites, we have a minimum of 2 visits that are spaced approximately 20 years apart. So a pile of plots that occurred in the mid-90s, and then another pile of revisits of those same plots around the 2010s, let's say. And these are all measured using the same protocol, getting whole community measurements of all lichen species that occur there, in many cases with the same exact observer 20 years later that had observed them in the first visit. So what we can do with this repeat visit information is look for the signal of lichen change. Climate signal, and the tool that I use is something called fuzzy set ordination. So I'm happy to explain what this is. It's a bit of a mind bender, but at the end of the day, what it's giving you is a way to assess the variation in lichen changes. In other words, how species were gained and lost over time, as constrained by climate changes or as attributable to climate changes that were also observed. So we're looking specifically at changes in mean annual air temperature. So that's the warming signal, and changes in climatic moisture deficit, which is a drying signal, if that's present. So this fuzzy set ordination basically gives you a score that lets you assess that. So the results of that give you a map that looks something like this. And let me pause to explain briefly. What you're looking at is a map where each individual point represents one of the sites that I showed you previously. It's a plot that's been resampled, colored by the amount of warming that was actually observed at that plot over the 20-year period. And the distance in between plots here indicates their similarity or dissimilarity of the way that their community compositions changed, of the way that they gained and lost certain species. So more just like a map of cities gives you an indication of how distant those cities might be out in the real world. Here I'm giving you a map where the distance between points indicates their compositional change similarity. So for example, 2 points in this upper corner changed in a really similar manner. They also had similar degrees of warming. But let's say this point up in the corner versus this point way out here were vastly dissimilar in the way that their community changed. Another way to interpret this in terms of the axes that you're on, we're not looking for like a nice clean regression line or anything, just like a map of cities doesn't give you a regression line. What we're looking is the way that the climate change variables overlay onto the map. So here I'm showing you warming, which is how we interpret our first axis here, a warming gradient. And the second axis is attributable to drying or climatic moisture deficit. That's probably all a little bit confusing. I mean, I'll admit it's confusing to me a little bit, but why don't we look deeper inside the model and just say how much of the variation in lichen communities is attributable to climate changes? And what we find is that measure of fit to climate is about half of the variation in vegetation change, in lichen community change, is attributable to a combination of warming and drying. If we look at those axis scores in relation to the actual warming in this case, or drying in the right-hand panel, I'm showing you the scores I just showed you from axis 1 have a fairly linear relation to the actual change with a measure of fit of about 0.57. So like in ecology, that's like a pretty good measure of fit, and slightly lower for the drying axis, which is about 0.47. But both of these we consider fairly decent correlation of score of the fuzzy set scores versus the actual climate change. It tells us that we can attribute vegetation change to these climate changes with that effect size. When we map these out onto the landscape, so I'm showing you again the regional map where we have the Cascade Range plots here, and then a few Coast Range plots on the left, colored by the first axis scores. In other words, you can think of this as the warming related compositional changes. We see a lot of those high values are occurring in the Eastern Cascades. So these are often high desert or foothills sites in the Eastern Cascades. Compared to where actual warming occurred, so here hotter colors indicate more warming, cooler colors, moderate warming, and then the most purple colors, which are mainly on the coast, are actually cooling slightly. So there's correspondence between the observed warming versus our model. It's not a one-to-one correspondence, but fairly good. Likewise, we can do the same exercise for the second axis. So the fuzzy set axis 2 versus the actual observing that the observed drying that we saw at these plots. And again, we see a slightly less correspondence between where on the landscape the vegetation changes are occurring that are attributable to drying versus where the highest levels of drying actually occurred. So here, the highest levels are pretty much in the south of the study area, getting towards areas that are already quite dry, and then speckled throughout the western foothills of the Cascades. So the fact that we don't have a one-to-one correspondence in this model, if we plot the residuals from this model, if you think of the residual as like the unexplained variation that we weren't able to capture, the unexplained signal that we weren't able to explain, what that's telling us is that there are other drivers involved, other drivers of vegetation change that are not just the climate variables that we measured. What could those drivers be? That is what leads me to my fourth and final narrative here, the interaction between climate changes and air quality. So climate as like this large scale global change atmospheric process versus air pollution and air quality that is often a pretty local process. How do they intersect? With lichens, we find that many species are correlated in the way that they respond to multiple different kinds of stress. So that could be either a liability or an asset if we're able to disentangle the two signals there. So for example, at low levels of air pollution, you'll find a set of species that also tend to occupy cooler or moister climate conditions. And you get species turnover as you proceed up the pollution or climate gradient, where at the highest levels of air pollution, those species that tend to be able to tolerate those harsh conditions also tend to be the species that tolerate hotter or drier climate conditions. So again, if we can disentangle these two signals of pollution and climate, we can use that to our benefit. We use an approach to model the air pollution signal that's called an air scores approach. So an air score is an average of all the species that occur at a site of their tolerances with respect to pollution, to nitrogen deposition. So for example, if I had 3 different sites and each of them with 3 different species, we know that different species have different tolerances. So site 1 occupied by a trio of intolerant species, so ones that are quite sensitive to pollution. If we average those tolerances, we get a fairly low air score, right? So if I averaged A, B, and C's value, maybe it's around 2.5 or so. Site 2 is more intermediate with a mixture of intermediately tolerant species. And site 3 would have a much higher air score because it's occupied by 3 species that are quite tolerant of high pollution conditions based on its community composition. So what this air score gives us is a way to rank or compare sites based on apparent deposition as seen by or reflected by the lichen response. So based on the community composition, we can assign each site a score on the deposition gradient. That is the meaning of air scores. And when we map those across the landscape, here again at each of those forest inventory lichen plots is a single point colored by its nitrogen air score. Relatively purple or cool sites are fairly low air scores, and hotter colors are higher air scores. If I gave you this map and you thought it was a map of actual observed air pollution, you would be forgiven for being mistaken because the way that the lichen response maps onto the deposition that we observed is a fairly good relationship. We have a lot of air pollution and nitrogen deposition in areas of the east. You can see that kind of lighting up, as well as the Central Valley of California. You can perhaps even make out the Willamette-Puget Trough over here and the Columbia River Gorge splitting Oregon and Washington. So that's good, that's as desired. We want lichen responses, if they're good bioindicators of pollution, to reflect that. We can also model air scores as a joint function or an interaction of not just air quality, but its interaction with climate. So here I'm showing you all of the nationwide air scores, the thousands of plots we have, each with its air score, a regression of those air scores on the vertical axis across the nitrogen deposition gradient. Also partitioned here by climate, by maximum August temperature, so the hottest summertime temperatures that these sites encounter. And what we see here is that if you are at a hotter site, this upper kind of gold line, you have a predicted higher air score than a site at the same deposition level that just happens to be cooler. So we can use that information to our benefit to tell us about where we find collections of species that will be trending towards hotter or more air polluted atmospheric conditions that are at the high end of the air score gradient, or at the low end, those species that are that make up a mixture of cool and clean air. Let me show you this in 3 dimensions instead of this 2-dimensional slice that I'm showing you. So this is that same exact regression, just plotted in 3 dimensions so that you can get a sense of the contours of that. Again, the vertical axis here is increasingly polluted sites from the air scores and decreasingly as you go down vertically, against the nitrogen deposition gradient here on the right, and increasing temperatures as we proceed away from the viewer to the left. Well, based on this interaction, what if I had a site that I knew was at a given deposition and climate condition, under a certain condition? I knew the values for that particular site. We can ask the question of what is the trajectory of that site on this air score surface if I were to apply different levels of nitrogen deposition or warming or cooling on the thermal axis. So we can run through scenarios that give us those trajectories. What if from our starting point, we add more nitrogen? So we are polluting the system even more than it is, and climate change is proceeding as expected, adding 2 or 3 or 4 degrees Celsius this way. So the red line here you can see is the steepest increase up these contours. It means that steep increase means that we're going to see the steepest change or the fastest rate of change in the lichen communities away from mixtures of species that are clean and cool tolerant and towards ones that are tolerant or indicative of hotter, drier, more polluted areas. Likewise, if we have a do-nothing strategy, we see climate change proceeds, but we haven't changed nitrogen deposition, we get a slight increase in that air score. So a community change that proceeds perhaps not as quickly. If we reduce nitrogen, again, a local solution that we can reduce non-greenhouse gas emissions, we can stay fairly constant across the contour. But if we both reduce local deposition of non-greenhouse gas emissions and society collectively finds a way to reduce greenhouse gases that are climate forcing, we find that the reduction of those non-greenhouse gases in combination with the societal improvements can potentially offset what we expect climate changes to occur. So what this says is we have a potential local solution to a larger, more complicated, multi-level societal issue. Okay, I just want to briefly summarize the 4 stories that I shared with you and then open things up for discussion if you would like. I showed you in the first study that individual performance declines as you progressively add warming. in a way that leaves species species susceptible to change, which relates to study number 2 that says future levels of warming may increase the vulnerability of communities to community shifts, changes in the identity of species, which we actually observed in study 3, where we related changes in climate to observed lichen changes. And then finally wrapped up how we can use the relationship between climate and air quality to help us offset potential climate effects. I do want to acknowledge all the many contributors to this work, since it's weaving a few different things together, but specifically funding from the Forest Health Protection Program, from the Forest Inventory and Analysis Program, and my current employer, the Air Resource Management Program, all of the Forest Service. So if we have time for questions or comments, I'd be happy to take them, perhaps as Lalita directs towards me.
Thanks, Rob. That was awesome. So helpful after reading your paper to kind of dig in a little bit and hear more about all of these different aspects. And the storytelling quality of your presentation was really great. It really put a nice storyline through the whole thing. So I am going to check the Q&A here. Misha, one of my students, wants to know a little bit about your background. So, and I had some other questions from my students that I read in their study question responses about this as well. And a lot of it stems, I think, from an interest in finding out, how do you get involved in a career like this? Misha's specific question was, how did you get into statistics?
I find that you just kind of meet certain people in your journey through life, so that may just lead you down a totally separate branch that you never knew was there. So for me, it was just a combination of meeting those people who happen to be trained or capable or well-versed in those things, but also specifically for the modeling and statistics, the need with this global change issue to detect like a really subtle signal amidst a really noisy ecological system. So while if you say ecology to many people, they may think you go out, you look at plants, you maybe take pictures of them, maybe press them in a plant press, and that's where it may stop in their mind. For me, ecology is like, why are these things occurring there? And how can we attribute changes in them to can we attribute changes, and then how do we do that? And so the modeling tools are useful for getting those really subtle signals to make a claim about them. So I'm not sure if that answers the question though.
That's great, thank you. Yeah, and some of my other students were asking similarly, like what came first, your interest in, bryophytes and lichens and ecology, or was it the climate that you studied in grad school? So how did that come together for you?
Yeah, I think I've always been interested in, let's say, forest vegetation. And then again, through just meeting the right people, got the specialization in lichens and bryophytes. And they turn out to be very useful for global change detection, or they have certain uses. So I don't know which was the chicken and which was the egg, but I view like the modeling and statistics and like the highly structured academic side of things as being the hammer for the nail. So the nail is like the global change issue. That's the question that came first.
Great, thank you. Okay, Jason Holluck is asking, are the Midwest and Florida too warm for lichen, or is there another reason there's no data there?
So you were, I presume, talking about the map of plots. So the Minnesota has FIA plots that I've used, and that's also where the experimental work happened. So Florida, the gaps that you see on the FIA map are there for two different reasons. One, in the central US and in the Great Plains states, there are forests and woodlands, but not very many of them. So the strategic decision was made years and years ago at the FIA level to not sample lichens in those areas. They do sample trees and understory veg in those sites. Florida and let me think, Tennessee, a few of the other eastern states basically, for the managers who controlled the purse strings, the budgets there did not deem it a priority to buy into it in the 90s and 2000s when the sampling was really happening. So there could be supplemental studies, so off-grid or off-frame studies that could happen there, but simply haven't happened yet. Florida is definitely going to be an area where lichen change occurs, as in Minnesota, as we showed. So changes will occur everywhere, right? But we want to know what's the rate of change, what's the identity of species that will change. There's a lot more questions.
Thank you. Aiden would like to know, what follow-up study would you want to do if you could do anything?
Oh, there's a few things. I mean, I have follow-up studies right now that I'm trying to brainstorm, but one thing that's on my mind is this concept of climate migration. So most of what I've showed you today is how climate will restrict the ability of lichens or species or communities to occur. But can lichens track suitable climates? Can they escape harsh environments and track suitable ones? So I have some, I'm versing myself in something crazy called circuit theory that basically tries to model movement across a landscape in the same way that electrons would move across a sheet of metal or an electronic landscape. But what you can do with that is constrain it by rates of climate change, by the availability of forested areas that it could inhabit. So that's one direction I'm thinking.
That's really cool. Thank you. We've got lots of great questions coming up. So
Good.
Sasha would like to know, are there any other organisms that have been researched in terms of community vulnerability in response to climate change, such as mosses, insects, birds?
One of the best ones for vulnerability has been birds. The bird people are, I guess you'd say, ahead of the curve on a lot of the climate change forecasting. So that's one. Definitely for forest plants, both for understory vegetation, especially I can think of a few studies in Eastern Canada and the Eastern US. I believe not so much for trees, but kind of addressing the last question we just had, one of my current projects is looking at intersecting vulnerability for different morpho groups, if you think of them. So basically trees, shrubs, herbs, and lichens, do we see vulnerability that co-occurs in the same way geographically? And do we see groups that are changing at different rates or expected to change at different rates? So that's part of basically the contributions from the Forest Health Program have been to look at the intersecting stressors, and they care mainly about trees, I would say, but about all vegetation, to look at ways that those environmental stressors may increase pest and pathogen risk. So insect and disease outbreaks may occur more frequently where trees are stressed. So that's also ongoing work and I guess a different group beyond lichens.
That's cool. Okay, great. So next up we have Brian, and he's wondering, I noticed an absence, oh, maybe this is a similar question that you already answered. I noticed an absence of data in middle states. So that one's been addressed. Let's move on to Emma. In the black spruce bog warming regimes, has there been any looking at changes in the secondary metabolite production of lichens?
No, although my colleagues up there have interest in that. I think that's an interesting angle because they are directly involved in the stress response. So
yeah,
I guess I won't get into the details of that, but that's one of the reasons why fungi in general, not just lichenized fungi, have sort of correlated stress responses, because phototolerance, tolerance to high light, is often correlated with heat tolerance, is often correlated with pollution tolerance. So like the same metabolites and basically the genetics that make them function similarly. So I don't have information on that or current plans to do that, but my colleagues at University of Minnesota do.
Great. While we're talking about that experiment, I did have a related question. So I thought that the individual experiments were really interesting. I wonder what you think about the transplants themselves and how, climate change would happen over a longer period of time than just kind of bringing a lichen from an individual from outside inside of a chamber, right? And so do you think that if those lichens, if they were more slowly, I mean, maybe it was enough years to see, but would there be some adaptation that could happen, or acclimatization that the samples might have under slower warming that would lead to them having less of the negative results that you were seeing, do you think?
I would suspect yes. So there was a slight ramp up period over a period of, I believe, 1 to 2 months as they were turning on the treatments. And actually the below ground, the deep peat heating occurred almost a year before the atmospheric heating did. So there was a little bit of a ramp up there. But I suspect that what you're saying is if there's a more gradual, perhaps more realistic approximation of warming, that there might be time for adaptation, for acclimation, for the photobiont switching I mentioned, that could be advantageous. That being said, the fact that you can get like a 20% positive growth rate to 20% negative in over 365 days says that they're responsive. That's like a rapid turnaround time. And the fact that we don't see recovery over the 5-year time span. In fact, if anything, we see deeper responses over that time, where the harshest treatments are seeing continued persistent wasting of lichen individuals. That maybe that's not going, that might be a strike against that, I guess. I do have designs to do a recovery of function experiment, taking those most impacted individuals and putting them back into ambient conditions to see, man, did we really kill them? Are they functionally dead? Or might there be that capacity, for example, under future climates that fluctuate between a really harsh time and then go back into periods of moderate conditions?
Very interesting. Thank you. Lauren asks, how do you navigate collecting data in environments with an abundance of micro and novel climates? Which regions do you observe these to occur most in?
So if I think, so I'm using a massive data set when I look at the FIA data across the US. Clearly, I haven't gone to all those. Microclimates are definitely important for lichen communities. If we were interested in, if I went out and measured, let's say 5 or 10 sites, I would definitely have to account for microsite effects. The beauty of using the massive data set with 9,000 plots is that we can, at such large scales, we're actually representative of the full range of microsites, the full range of disturbance histories, of land ownerships, of anything else that's happened there. So that in a way, we're smoothing away some of the variation that you would attribute to microsites. But if you were doing something with a smaller number of sites, you would definitely have to account for that. I don't know the second part of your question. Where would microsites be most influential?
Which regions do you observe these to occur in most? Maybe she's thinking about the micro and novel climates.
Yeah, so for microhabitats, I guess I would suspect, I don't know for sure, but I would suspect if you had an area that was highly mountainous, had a lot of rugged topography, a lot of little riparian pockets, that variety offers a variety of microsites and of microclimates. Novel climates, when I say that, what I mean is something that's unlike anything that we've seen in our history as human beings, and that these plants probably haven't seen in several million years if they've encountered that at all. So that's sort of the tantalizing promise of the forecasting is, can we make an accurate, unbiased forecast to conditions that we've never seen, and that this vegetation may have unknown responses to, based on what we do know about their responses. So that's the challenge right there.
Thanks, Rob. That's great. Wow, lots of great questions in here. I hope we can get to them. We only have about 5 minutes left. Asaf asks, is there a practical use to growing lichens intentionally to help continuously monitor the local environments?
Yes, certainly. So people have used transplants to basically do a common garden experiment. or a reciprocal transplant experiment, where because you can transplant them fairly easily, say you have a clean air site and you want to assess a pollution gradient, you would collect a bunch of individuals from there and then distribute them along the gradient to be, I think people call them sentinels, ecosystem sentinels, if you want to think of that. So the answer is yes, common garden experiments and reciprocal transplants can be done, and probably should be done for climate gradients and measured repeatedly over time.
Great. Lauren asks, do you think it would be, this is another Lauren, do you think it would be possible or beneficial to attempt to mimic the bioindicator properties of epiphytic lichens in a kind of technology? Ooh, this is getting kind of sci-fi, which could become an even better and more sustainable indicator of climate change and air pollution?
Like nanotechnology, that would be neat. Yeah, I guess that's, certainly their climate sensors exist, and I guess I'm just making the claim, like, here we have biological sensors that, yeah, we need to do the modeling, we need to make adjustments to account for other things that are happening, but the fact that they are distributed basically on every tree and shrub out there, if you start looking, if we know how to read them, they provide that to us. Yeah, I don't know. I would have to think about what engineering or technology you could do with that. Maybe maybe was it Lauren has ideas that they'd be willing to share.
Yeah, awesome. Okay, Lily asks, in your 2002 study on climate niche limits, maybe it was the 2020 study, and community vulnerability, it was suggested that host species like trees would have potentially dramatic changes in range or composition, which would then affect lichen communities. Has this impact been studied? What could or would lichen transition to different hosts, or simply would they transition to different hosts, or would those lichens be lost? Would transition to different hosts affect community composition?
Let's see. So I believe in the paper, I know we cite at least one 2016 study that Greenwood et al., 2016 if I recall, that looked at tree line advance in a mountain system and community changes and inferred rates of colonization through a tree line ecotone. And so that is evident, and found mismatch between tree colonization and lichen colonization. So that's evidence that yes, if you see larger, not just that local mountain, but larger biogeographic shifts in the available tree species, like if you have nothing to grow on as a lichen, you have nothing to grow on. So the actual physical substrate would definitely impact community composition, or at least abundance of lichens. The question of host specificity is mixed up with that, but somewhat different, meaning that does one lichen only occur on one tree species? We find that's usually not the case. Actually, the vast majority of times it's not the case, but rather it's the physical characteristics of the bark that often tend to be most important. So, and that often cleaves along lines of hardwoods versus conifers. So many species can use like, a fir or a Doug fir or a cedar, but they won't occur on an oak, for example, and vice versa. So that's sort of a related but separate question. But yeah, I'd expect shifts in the trees will impact shifts in the lichens.
Great. Well, we are at 12, but we have a few more questions. I think maybe your presentation has your email in it if folks had any additional questions. I'm not sure.
I could put that back up if you like.
I also could let students know. But I guess this is time to thank you so much. If we had the clap feature on, we could all clap for you. Thank you so much, Rob. This has been an awesome talk, and I think all of the attendees got a lot out of it.
Well, again, I appreciate the invitation, and it was my pleasure entirely. Thank you so much.